So hello, Francesco Pignatti. Very happy to have you here by Visio. So to present yourself quickly, you're a medical doctor. You have also training in biostatistics, and you're working at the EMA, so the European Medicine Agency, in the Oncology, Hematology, and Diagnostics department, mostly on the approval of medical products. So we are very happy to have you, and we have a few questions on your activities. So first, could you present your activities at the EMA, please?
Yes, hi, and thank you very much for the opportunity to be here. I've been at the agency for almost 25 years now, and I've pretty much always worked in the office of oncology, which is an office which supports our scientific committee. Our scientific committee, I mean mainly our Committee for Human Medicinal Products. This is a committee that evaluates new drug applications, for example, and then decides whether they should be approved or not, and what should be the label, what should be the therapeutic indication, what should be additional studies to be conducted post-approval, and so on. And at the agency, we have a small team of scientists who support this committee in their decisions. Aside from these activities, I'm very interested in all aspects of cancer research and methodology, and also in benefit risk assessment, which is a central part of our decisions.
Could you explain to us, Dr. Pignatti, how the Darwin project could help on this situation of approval of compounds or even repurposing of compounds? Well,
thank you. It's an excellent question, and you're right. This is a big project for the agency, and we have invested a lot. Because we've realized over the years that real-world evidence is evidence that can be helpful in many situations. And if we just look at our pharmacovigilance databases and so on, we see that a lot of this type of data is necessary and useful. Where we relatively lack experience and is a bit of a question mark is if well-curated evidence could be used to replace randomized clinical trials. In situations where you're lacking the ability to conduct randomized controlled trials, then it would be extremely convenient to be able to get this information in a different way, for example, through real-world evidence, and build your control arms like that, and have reliable methodologies that allow you to make some conclusions. And that's the big question mark. That's the big challenge. I think the jury is still out. There are many who see this as a phenomenal opportunity and perhaps the future, where randomized controlled trials will be something of the past. And others who are much more skeptical, that say randomized controlled trials have a specific purpose, and it is not through this type of methodology that you can replace them. I think this question is still open. We'll see how it goes. My personal view is that randomized controlled trials are there to stay. They provide a certain level of methodological completeness, rigor, that is difficult to replace with data collected in a different way. But I think that randomized controlled trials is a broad term. There are many types of randomized controlled trials, and some of these are nowadays called pragmatic trials, where we are more aware of the difference between the very selected populations that one often sees in randomized controlled trials and the population that is then targeted in real life, and that we need to bring these together. So that's one very important topic that for me will remove many of the objections, let's say, to randomized controlled trials. And for the rest, I suspect that we will have to say in situations like where single arm trials are the only thing that we can do, I suspect that we will say that, well, carefully conducted, externally controlled trials using real world data are probably better than nothing, than just informal comparison to some sort of old paper in the literature. I think carefully conducted real world data is in a much better position. But uh, it is known that there are so many challenges that most likely it will not achieve the, let's say, the coverage of questions of randomized controlled trial, but will have to be reserved for certain questions which are perhaps considered more secondary or only for situations where the differences are very, very extreme, and so on. So we will have to find the proper role of these real world evidence studies. It will not be necessary to replace randomized controlled trial, but there will be very useful roles, no doubt.
Yeah, very interesting and very clear. I know in Primrose there will be some development of methods for such comparison of single arms compared to real-world data. Could you explain to us some of the methods that could be used for such comparison,
Yeah,
please?
I'm not an expert in this,
Yeah.
unfortunately, but I find it fascinating. And one of the problems with all these methods is that they rely on certain assumptions about what you
Mm,
know and what you don't know. And then you can work under these scenarios, and often this is enough to provide you some sort of a bandwidth of how wrong can you be under the different assumptions. And sometimes this is enough. But most of the time, because you don't know what you don't know, it's very difficult to test those assumptions.
mm,
And now if we bring these methods to our, let's say, rare disease, rare cancer type, single mutation, very rare, and so on, those are often the settings where we don't know a lot, and we don't have a big data set of controlled, so carefully collected data in an external population. For example, it might be that the association with the biomarker has only just been discovered, and then you don't even have these external controls. You don't know what are the prognostic factors in that particular disease. So in a sense, sadly, this whole real world evidence is going to be more difficult to use where
mm,
we need it most. Those
mm.
situations where the diseases are rarest and we have less information. So really the jury is still out about how useful this is all going to be. My bet is that this is going to be much better than nothing, but it will be a, a difficult, let's say, an upward learning curve.
Yeah. Okay. Yeah. Very interesting to have your view on that, and I think the challenge will be for the methodologies to balance those limited knowledge we can compare our study to. So that's very interesting. Um, Concerning those type of data, so what type of data will be used in the Darwin project?
So the Darwin project is at its beginning now. So they're creating this incredibly complex but efficient system of federated databases that will be able to answer questions. And I think they have planned to answer a handful of questions for 2023 to test the system. It will be questions about drug utilization and safety. But the whole system is planning to be up and running in, I think, in a matter of just a few years, to an
Mm,
extent that it will be able to answer hundreds of questions in a year. And so we're looking at the questions that our committees will generate um, around the drugs that I suspect are approved, or we don't just have marketing authorizations. We have a number of other decisions, for example, about orphan designations, looking at prevalence of diseases and so on. So there is a whole catalog of questions that can be answered. But I think the main purpose will be to address questions by our scientific committees.
Happy to see some of the questions and hopefully the results. Maybe a more specific question. Is there a registry at the EMA for the different approval and the indication of approval, as for example the FDA has? So that's one of the type of data that could be very valuable for many research application. Is there such data?
Yeah, I think it's an excellent question, and I'm not sure what is the FDA database, so I cannot compare. But I think we strive for putting all information available on our websites in structured format. So even if it might not be, let's say, the format of an Excel spreadsheet or something that synthetic, but I am told that with today's algorithms, it's very easy to sort of extract information from our structured publication type. Now, I'm sure this can be improved, don't get me wrong. But let's say the step one is there of having a maximum of information published about all of the drugs we approve and the drugs we reject, which is also an interesting source of information.
Okay, thank you very much, Francesco Pignatti. I think we covered a lot of topics of great interest, and I hope this would also be of interest for our audience. So thanks a lot for your answers.
It's my pleasure. Thank you.